All right, with no further ado, I want to introduce you to today's speaker, Dr. Alex Kwan from Cornell University. So Alex was trained in engineering physics, and in 2009, Alex received PhD in applied physics from Cornell University, where he was working on nonlinear optical microscopy in the laboratory of Watt Webb. And from 2009 and 2013, Alex did a postdoc in neuroscience in the lab of Yang Den, where he worked on neuronal circuits. After that, he became associate professor at the Department of Psychiatry at Yale School of Medicine. I think this is where his psychoactive drug career started, perhaps. And in summer last year, Alex moved his lab to Cornell University to Meinig School of Biomedical Engineering. And here is where we are today. Alex is becoming one of the leading figures in studying both structure and function of neural circuits and how they're affected by a variety of psychoactive drugs.
So I want to start by thanking Meng for being a wonderful host for the last 2 days, and she also really gave a great introduction, and to her and also to the rest of the organizing committee for inviting me. First time being in Berlin, I really enjoy it. I had curryverse a couple times. I went to several climbing gyms. It's been really wonderful, really enjoyed it. So yeah, I also talked to a number of research labs here. I think that's very exciting, a lot of psychedelic work happening here. And I'm glad to have this time to tell you some of the latest research from my own laboratory. I will start with disclosures. So I consult or advise for a couple of companies. This includes Freedom Biosciences, Empire Neuroscience, Silo, and Biohaven Pharmaceuticals. That being said, all of the research that I'll show you today are supported by grant support. So this includes 2 grants from the NIMH, which is NIH in the United States. We have some money from Brain Initiative to advance optical microscopy, and a grant from One Mind Foundation. The student also received some support from NIDA and Source Research Foundation. And finally, all of the drug work that I show you, we got the compound through a supply program from the Usona Institute, which is a nonprofit organization. This is my lab. So we have about 10 people, 5 postdoc and 5 graduate students. And I'll try to highlight their contribution as I go along, what they individually contribute to each of the projects that I'll show you. So in a nutshell, very broadly speaking, my lab, we're very interested in psychoactive drugs. We're very curious how these molecules can cross the blood-brain barrier, enter the brain, changes structure and function of neural circuits, and then lead to some behavioral changes. And we're particularly interested in behavioral changes that might be beneficial for treating mental illnesses. And then in this regard, we think our work can be useful because we ultimately want to predict how new compounds and new drugs might work. We think that if we can predict how they might act on neural circuit, we can also predict what the behavioral changes might be. Another goal of the lab is, once we understand what some of the neural circuits are involved, being in an engineering department is also in our mind to always think about how we might be able to engineer certain circuit pathways. So we think about ways that maybe if you can stimulate the circuit, maybe at some point you can replace the drug, or maybe even augment some of the drug effect that we see. So we think these are quite relevant for mental illnesses, and this is really some of the long-term vision and goal for the lab. Within this context, we have been studying psilocybin in the last several years. So the chemical structure of psilocybin is there on the left, and in the body it's rapidly metabolized into psilocin, which is what gets into the brain, binds to serotonin receptors, and causes a range of interesting behavioral effects. Both psilocybin and psilocin can be found in a so-called magic mushroom, there's a photo here on the right. There are 200 plus species of these psilocybe mushroom, which is actually found all over the world. This particular one is Psilocybe mexicana, which is actually the ones where psilocybin was first chemically isolated. We wrote a couple of reviews paper over the last year. If you're interested in this topic broadly, I encourage you to look at it. The current biology paper, we wrote it for lay audience, and then in a simpler language. And then the other one in Nature Neuroscience, we try to bridge different levels from molecular to cellular to systems and to network, trying to get a more comprehensive view on what we currently know about psychedelics. And you might already know, since you're in this lecture series about psychedelic, what these drugs do. But just a way to illustrate this, I like to give this example. So this is a work done in Denmark by a Danish researcher, where they administer psilocybin to adult participants. And this is a female adult. And then afterwards, they ask them to describe that experience. So here on the left is what the woman drew about that acute experience. You can see it's very visual, very colorful, very vivid. On the right is what she wrote, and I'll read it out aloud. So this, again, after psilocybin. My inner vision became the universe, filled with colorful waterfalls, glistening like stars, not bound by gravity. They floated in the air and folded around each other, totally quiet. The streams were infinite and beautiful. So you can see these are very powerful compounds. And I picked this passage because it illustrates several key points of the behavioral effects. One is the change in perception. You can see that a lot of it could be quite visual, particularly for psilocybin and other drugs like LSD. There's also emotional changes as well. Some people describe it as mystical-like experience and general like connectedness with the other people or with the world. So these are acute effects. So in human, in oral consumption, it lasts for about 4 to 6 hours. But as you all know, the psychedelics also receiving a lot of interest in psychiatry now because of its potential use for treating mental illnesses. So one good example, I think, is these results from phase 2 clinical trials. So there are now a number of clinical trials being ran or have been run, some in UK and Johns Hopkins by Compass Pathway, as well as Uzona Institute. I think this particular one is from the UK. You can see that with 2 doses of psilocybin, there's a fairly significant decrease in the depressive symptom. So these are patients with depression. And then there's also, the reduction is quite sustained as well, which is quite striking. And this is notable if you compare to placebo or SSRI, in this case fluoxetine, which is taken daily. Again, you can see how fast and how sustained the effect is. So this has created a lot of excitement in the field of psychiatry, because if this can be proven again in larger phase 3 clinical trial, it could really transform treatment for the people that respond to psychedelic. So given that there's a lot of excitement, there's also a lot of news around it. I think it's now really in popular awareness. So here's a couple of headlines, a headline in New York Times, cover story in Newsweek. You might know Prince Harry talk about it on 60 Minutes, about how he uses psilocybin to deal with grief. And then there are also books now, right? So this is probably one of the most popular books, How to Change Your Mind by Michael Pollan, describing his own experience on psychedelic, as well as some history and society issues. So my lab, we're not so much care about how to change your mind, but we are very interested in how these drugs can change your mind. So understanding the neurobiology and the neural mechanism behind these drugs is something of great interest to us, and that's what I'll tell you about today. So we don't know a whole lot about what these drugs do, but there are some hints and some potential starting points. And this comes from knowledge about other compounds that are used before to treat depression. It's been known through a couple decades of work from people like Ron Duman, Lisa Montega, and others, that many of these drugs that are effective for treating depression acts on dendritic spines. So here is a cartoon of a pyramidal cell in the cortex. You can see a cell body in the beautiful dendritic tree. Each of those little knobs is a dendritic spine where the neuron receives excitatory input. The dendrite is very important. It serves to receive, integrate, and process these inputs. And then through earlier work, it's also known that some of the regions important for these effects are located in the medial frontal cortex of the mouse, which probably correspond to the anterior cingulate cortex in humans, among other areas that are important. So this is also where my lab started our work in this area. So for today's talk, I will first go through actually some published results, but quite quickly, just to lay the foundation again on where we started. And then I do want to move on to some newer results where we now try to go from the initial study around the dendritic spines, but into broader circuitry and brain pathways that we think are quite important for the action of psilocybin. So when we started, we didn't know what we were getting into, so we want to make sure that we have the right compound and that we're doing the correct thing. So one of the classic assay for looking at psychedelic effect is the head twitch response. So this was work done by PhD student Clara Liao and also postdoc Ian Gregg. And what we did is we mounted a high-speed camera on the top of a ceiling of a box. And you can just look at the mouse's behavior, and you can see the mouse on the left that receive 1 milligram per kilogram of psilocybin have the rapid side-to-side head movement. And this is well known from the 60s that animals, particularly mice and rodents, that receive psychedelic would exhibit these side-to-side head movement. So what we did very first is for Clara and Ian to map a dose-response curve. So we map, well, I think over 60 mice here of both sexes. You can see there's a clear inflection point at about 1 milligram per kilogram where you can see the drug can get into the brain and really elicit robust head twitch response. And this is the dose of the drug that we'll use, and I'll show for the rest of the talk. And I should mention that, you can do this with video, and then there are also other people. I think Javier Gonzalez Meso has gave a talk here. There's also some really nice way to use magnetic ear tag to measure head twitch response. It's a very standard assay. And then we also have put up some manual on how to set up some of these boxes, some of which actually based on Javier's design, but it's on GitHub if anybody want to build their own. It's quite cheap and easy to build. And most of the lab, in my lab though, we're interested in looking and visualizing neural circuits. And generally what we do is we work with head fixed mice. So here on the left, you can see the mouse is awake, but it's head fixed on its head, so we can do cellular level imaging and recordings. We use a two-photon microscope to visualize the neuron structure. Again, we're focusing on this medial frontal area. So this is the anterior cingulate CG1 and medial M2 area in the brain of the mouse. And we either load the fluorescent protein using a transgenic mice, in this case 5-1-GFPM, if we want to do structural imaging. Sometimes we use viruses. And then finally, we replace a skull with a piece of glass window, so for optical transmission. If you come to the lab, this is the kind of image that you could see under the microscope. This is with the two-photon imaging. These are GFP expressing neurons. We're just going into the cortex in layer 1, and you can see these really nice, again, beautiful dendritic branches. And hopefully you can also see the little knobs on there, right? Those are the dendritic spines where the neuron receive most of its excitatory input onto the cell. So in this early study, again, this already published, Ling Xiao, who's a postdoc in the lab, tracked these dendritic spines over time, and we were very curious about what is the action of psilocybin. What we reported is that a single dose of psilocybin can lead to robust structural remodeling of these dendritic spines. So you can see more of it. If you look at the spine density, you can see an increase of 10% on day 1. Most remarkable, I think, is how sustained it is. So we look at day 34, and you can still see that increase, the difference between the drug and the saline. Another thing that we observe is that the spine head width, the size of the spine also increased. But it has a different time course here, which is quite interesting. You can see that it increased by about 10%, and then it goes back down on day 30. So the size of the spine usually indicative of the strength of that excitatory synapse. So these things represent different indicator of different aspect of the excitatory transmission. I want to specifically highlight that because actually that will come up later in terms of the spine density, spine size. Once we go into different cell type, we also see some interesting differences there. I get this question sometime, right? But like, are these structural changes also functional? Do they mean anything to the neuron? So in this case, Pasha Davodian, who's an MD PhD student in the lab, he made slice electrophysiology 24 hours after drug administration. And these are miniaturized EPSC. And what you can see is that compared between saline and psilocybin, and for both sexes, you can see there's a clear effect on the frequency of these minis. And then on amplitude, there seems to be an increase, although the p-value is 0.06. But this suggests collectively that whatever we see structurally, also you can find correspondence changes in excitatory transmission in these neurons in this area. Okay, so this is sort of the end of the first part. I just want to set the stage on where we came from, and then I'll show you some results that we built on these studies. But just thinking about, why are these important? Why do we care about neurons in the spines and what the drugs do to them? One, thing that we know about depression is that there's a number of evidence suggesting there's synaptic atrophy or spine loss, synapse loss in the frontal cortex. So potentially the drugs, by potentiating or increasing the number of excitatory synapses in these areas, could reverse some of these atrophy. And then another, I think, exciting possibility is that one could also ask, when do circuits form new spines? And that happens when the animals tend to learn new tasks, at least in adult animals. So it's possible that these new spines or potentiation of existing spine could help with long-term integration of new experiences, particularly those that one might encounter during the drug dosing. Okay, so that again was the first part, and I think some of these early study was exciting, and we were very happy to see these quite drastic change in the dendritic spines. But dendritic spines are in pyramidal neurons, and they're obviously embedded in local and long-range circuits. So we want to start thinking about what else is going on in the brain when we have psychedelic on board. So in thinking about this, there are a couple of ways one can go about doing this, right? And this is actually a jigsaw puzzle done by my daughter, 9 years old. The two approaches you can do solve a jigsaw puzzle. One is if you know that piece on the top left is the piece you want, in our case maybe dendritic spines and pyramidal neurons, and you can maybe find pieces of similar color if you know that's what you're looking for. Another way to do it, a little slower way, but you can find the pieces that connects to it. So that's sort of exactly what I'll show you, the two different approaches we took to move this study along. And I don't think there's a good or bad, right? They have the pros and cons. On the left is a high throughput method. I think is very useful for screening drugs, for drug discovery, asking if certain drugs behave similarly and getting an idea on what brain regions might be important. On the right, I think is something that, detailed circuit mapping, something that can really reveal some basic fundamental neurobiology. So this is the two ways we go about trying to understand the circuitry involved. So for the high throughput method, Pasha Devodian, who's an MD PhD student, he did light sheet microscopy. So how this works is we injected the mice with either psilocybin or ketamine, which is another fast-acting antidepressant, or saline. We estimated that the drug probably takes on the order of 30 minutes to reach its peak activity. And then we know from prior studies, CFOS take about 1.5 hours to express fully. We collect the brain at that time point, clear the brain, and then immunolabel them. In this case, we label CFOS, which is a activity-dependent transcription factor. So it's activity-dependent kind of plasticity-related gene. And then we put the clear brain for light sheet microscopy as a way to sample the whole brain rapidly. This is the kind of image that you can get from CFOS imaging. It's also, I think, quite beautiful, just different kinds of image. But light sheet is a horizontal sheet, right? So you can see this brain now is a horizontal section. This is one sheet through the brain. And if you magnify the cortical region, you can really see the puncta corresponding to the CFOS stain. One advantage of CFOS is that it's nuclearly localized. So we apply a machine learning algorithm to automatically detect all of the nuclei that are CFOS positive in the different regions of the brain. Again, either after psilocybin, ketamine, or saline treatment. So this type of work leads to a very big data set. I don't assume you can see this, but this this table is also published. But what we did is we fit the brain to 316 different brain regions as defined by the Allen Brain Atlas. This is based on the common framework. And then we align it, and then we ask how does psilocybin and ketamine drive CFOS expression as a function relative to saline. What you can see is that ketamine and psilocybin activates many regions commonly. So regions like the place we image, for example, the ACAD, which is the dorsal anterior cingulate cortex, prelimbic cortex, primary visual cortex, retrosplenial cortex, medial dorsal thalamus, locus coeruleus, lateral habenula, costrum, and some of the amygdala region. So both of the drugs have some common region that they activate that share. And then some of the psilocybin-only region which we're quite interested in include insular cortex, which I think is actually quite fascinating given its role in some of the emotional processing. PAG, which is commonly associated with escape behavior. Dorsal raphe, this is quite well known that psilocybin and other psychedelics suppresses the activity of dorsal raphe. So this is actually a good check because we also see a substantial decrease in CFOS signal in the dorsal raphe. And then for ketamine, some of the regions that's specific for ketamine are infralimbic cortex and some of the hippocampal CA1 and CA3 fields. With a big data set, I think it's quite fun. You can do a number of things with it. One of the things that Pasha did is to take other bioinformatics data set and compare and ask what can predict the CFOS change on a whole brain level. So here, what he did is he extracted in situ hybridization, like transcript level, mRNA level, from a different brain area, and then run a correlation like what's shown on the bottom left here, whether some of the baseline transcript level, certain genes can predict the change in the CFOS level induced by a drug. So can we, based on gene expression, guess what the drug would do in terms of inducing these gene expression changes? Doing this analysis, we actually ran it through the 20,000 gene in the mouse genome that is available. And you can see on the top right there, the gray trace, the histogram, is all the genes, 20,000 or so. And then here, the lines are the serotonin and dopamine receptor. And you can see, maybe as you expect, the serotonin 2A receptor is quite good at 73rd percentile. Some of the other dopamine gene and serotonin gene is not as good in terms of correlating on a region-by-region basis. What's interesting is for this exploratory analysis, we also tried glutamate receptor, which turned out to be quite predictive. Some of the NMDA receptor subunit, like the GRIN2B and the GRIN2A, actually is above 90 percentile. Yeah, so we didn't expect that going in, although maybe now you think it's not that surprising. I just told you that the dendritic spines are quite important, right? So definitely some of the glutamatergic signaling is maybe involved as well. So the other thing that we've done now is with these kinds of more high throughput, faster approach, it's really hard to stop at 2 drugs. Once you do them, you start to want to do experiments on more drugs. So this is work from master student Farid Abouhab, where we now tested 8 different conditions, 8 mice each. So on the first one is called 6-fluoro-DET. I don't know any of you know this one, but this is an older non-hallucinogenic 2A agonist, which is actually tested in human, but it's non-hallucinogenic. Not clear if it's therapeutic. We also test fluoxetine, which is SSRI, either acute or chronic administration. 5-methoxy-DMT, which is another tryptamine, just like psilocybin, but shorter acting. And then ketamine, MDMA, psilocybin, as well as vehicle as a control. So we're still analyzing these data, but what I can tell you is that you can classify the drug very well. So here we're using regression-based analysis in machine learning approach to try to ask the computer to classify the different drugs based on the whole brain CFOS data. Farid is very careful in terms of trying to cross-validate and make sure that we're not overfitting. But you can see the classified performance, again, is quite good. So what's next is we're really trying to hone in now and ask what are some of the brain areas that's driving these classified performance in terms of at the whole brain CFOS level. So we think this is quite a promising approach if one want to ask, is this new drug, is it more similar to ketamine or is it more similar to psilocybin? If it's a bit different, then what might be the difference? Like what brain area might stood out as a difference in terms of the drug action? Okay. So that's, again, the first part of that jigsaw puzzle. Now on the second part, where we also want to look at circuitry, but from more of a tracing perspective, where we want to go slowly and ask what are some of the brain circuit involved. And one of the obvious, I think, follow-up question here is we show that dendritic spines in pyramidal neurons are responsive to psychedelic, but what about subtypes of pyramidal neurons, which I think also of big interest here. Several labs here look at different subtypes. So some of the major subtypes of pyramidal cells in the cortex include the intratelocephalic cell, so the IT neurons. So you can find these neurons in layer 2, 3, some of them in layer 5, also some even layer 6 of the cortex. And then the other major subclass is the pyramidal tract, PT, or sometimes also known as the ET neurons. And these neurons are predominantly in deep layer of layer 5. And you can see there's also other cellular properties that are quite different. You can see from this drawing, the PT cells tend to have a very elaborate dendritic tree, and the tuft is really fan out. It's also thicker in terms of the tuft, and there's also a lot of different electrical properties difference between these cell types. CT here is corticothalamic, which I won't talk too much about it. you can also see it tends not to have the dendrite up in layer 1, so presumably not the cell that we were imaging in terms of the dendrite spine changes. A key distinguishing feature of these cell type is that they have project to very different brain areas, which we think that if we can start understanding the cell type, we can also start breaking the circuit because we know what other downstream area might be important. So for this case, IT neurons both cell type project to ipsilateral striatum. The IT is unique in that it also go to contralateral side as well as other cortical region. So IT cell is responsible for most of the cortical connections, whereas the PT cell is strictly ipsilateral up in the helicephalon. It projects to striatum, some to thalamus, as well as to brainstem, including the pons, and some of it can even go to the spinal cord, be part of the pyramidal tract. So one might ask, what are the hints for why we think there could be some cell type differences in the response to psychedelic? So there has been a good amount of work looking at not psychedelic, but serotonin and the response of pyramidal neurons to serotonin neuromodulation. So give you one example. These are slice electrophysiology recording. You can see that when one wash on serotonin, this particular pyramidal cell depolarized. And then in the presence of serotonin 2A or 2C blocker, that depolarization is gone. So it seems to be dependent on the 2A and 2C receptor for this pyramidal cell. But when they patch onto other pyramidal cell in the slice, sometimes you can see other types of response. So here you see a cell that's actually hyperpolarized. And then in this case, though, if you apply serotonin 1A receptor blocker, you can see that effect is gone. So this suggests that pyramidal neurons respond heterogeneously. In this case, it's early on, they didn't know the cell types. And then they seem to involve mediated through different serotonin receptor subtypes that are expressed in these neurons. So this gives some hints that there could be potential cell type specific response. One thing that we did is we, together with MD PhD student Neil Savalia in the lab, we mined the single cell RNA sequencing data from Allen Institute. You can go and look at different cell types that identify and what transcripts are expressed in these cells. What we see is that, as other people have reported, a lot of these IT and PT cells co-express the 2A and 1A receptor. But if you look at what is the relative abundance of these things, IT neuron tends to have a little bit more of the 2A transcript relative to the 1A. Again, 2A is the one that makes it depolarized and excitatory. And you can see the PT cell is a little bit more equal. So there seems to be a preference towards the IT cell based on these kind of transcript data. It's suggestive. I mean, definitely it's not definitive, right? It's not protein level, it's not receptor level, but it's just transcript level. So based on this and some other evidence, which I won't mention, I think based on current evidence, it might be form a prediction that IT neurons could be more excited by psychedelics. So in the lab, we want to test that explicitly. So Ling Xiao, the postdoc, go ahead and identify PT and IT neuron, and we ran different types of experiments with it. So one way to identify these neurons is based on their long-range projections. So here, Ling Xiao injected retrogradally transport AAV, retro AAV into the pons, because we know these neurons uniquely project to the pons, and then inject pre-dependent GFP into the medial frontal cortex. And one good thing about this is you can titrate the virus that you inject into the pons, and you do a low titer, you can get very beautiful sparse labeling. In this case, we have GFP in a neuron that you can really fully visualize the dendritic tree. This is a single PT neuron. And if we do histology, you can also confirm that indeed they locate mostly in layer 5 in the medial frontal cortex. And if we now do IT neuron, we can use a similar strategy, but instead of injecting into the ipsilateral pons, we can inject into the contralateral striatum, again, based on the connection, labeled this type of pyramidal cell, IT cell. And accordingly, you can look at the lamina distribution and find that they're both in layer 2, 3, as well as layer 5, much more widely distributed. So one of the first questions we have after being labeled the PT and IT cell virally is, do they show different kinds of structural plasticity? So here, Ling Xiao have a very similar timeline as before, where we image for 2 times, give the mouse either psilocybin or saline, and then image for more times, except that we added a time point at the very end at 2 months later, 65 days later, because we were wondering whether it just keeps going and just sustain even longer. Again, you can see the quality. This is a PT cell. Again, the imaging is quite high, even though now it's virally mediated expression. It's still a good enough level of expression to really visualize the new and existing dendrite spines over time. What Ling Xiao found is that both cell types increase the spine density. So I don't know what we came in thinking, but end up both of them do. So you can see here that both PT and IT cell have elevated spine density, and both of them are actually also quite sustained. So we see these things out in their day 65. We don't know if it keeps going. At some point, I think you can also see for the IT cell, there started to be a dip even in the saline case, because sometimes the window quality get compromised, and also through repeated imaging, sometimes there's photo damage. It's really hard to keep going. But it seems like as far as we look, even out to 2 months, you can still see these new spines added and stay there. One thing that we do find is quite different between these cells is the spine size change. As I alluded to before, what you can see is the PT cell seems to potentiate much greater. And when we do the mixed effect model in statistics, we can see there's a cell type specific difference here, where the spine size grew for the PT neuron, whereas for the IT cell, the effect is not significant. So there does seem to be some cell type specificity. So both of them increase in terms of the spine number, but the PT is unique in that the spine also grew bigger. So, but what does that mean, right? Does it mean anything for behavior or physiology? So to answer this question, Ling Xiao did a chemogenetic experiment where we use chemogenetic tool to silence these neurons when the drug is administered. So we try to remove the neuron when the drug is being administered and look at behavioral consequences of that. To do that, now instead of sparse labeling, now we want to actually label a lot of cells. So here we leverage a couple of new transgenic lines from George Huang's lab. The FESF2 Cre-ER, which is an inducible Cre to label the layer 5 neurons, the PT cells, and then Plexin D1 Cre-ER, which is more specific to the IT neurons. And then to induce the Cre expression, we inject the mice with tamoxifen in order to drive. And then there's a Cre-dependent inhibitory dread expression. This is bilateral. So one of the behavior that we tested using this manipulation is the learned helplessness, which is a stress-related phenotype. So how this behavior works is that we would shock the mouse 2 times on 2 different sessions. And in this case, the mouse cannot escape. So we would shock, and then no matter where the mouse goes, it still gets shocked. So over time, some of the mouse learn to be helpless, meaning that they no longer try to escape. But some other mice are resilient, and they still try to escape and avoid the shock. So that's what we test on the day 3, the test session, right? And then in between, we give a dose of psilocybin or saline to see if the stress response is mediated by the drug. And then before the psilocybin now, we also give a dose of DZZ, which is a chemogenetic ligand to activate the inhibitory dread. Again, the idea here is that we want to remove, right, those PT and IT cell type in the medial prefrontal cortex before and during when the drug is administered. So on the right here, you can see the behavioral effects. We have shown this previously too, but a single dose of psilocybin is able to reduce the escape failure, meaning the mouse escape more, so they're less sensitive to the stress, the inescapable shock stress that they experienced before. So when we inactivate a PT neuron, we actually see that can largely abolish psilocybin's effect. So we're very excited to see this, where the drug seems to have quite a strong effect. Again, so it means that if we remove these frontal cortical neurons, at least for the learned helplessness task, right, psilocybin's effect seems to be largely diminished. And then we tested the IT neurons. For the IT neurons, you can see that it still shows a significant treatment effect, but there's no longer an effect interaction, right, between the chemogenetic probe and the drug effect. So here there's a treatment effect, but then you no longer see that you can block that effect if you just silence the IT neuron. So this data suggests that the PT neurons are important for psilocybin's effect on stress-related behavior. So one of the downsides for this behavior is that it's relatively acute. So in depression, people like longer stress period, right? Because this is a very short and maybe traumatic but short stress. So one of the things that Ling Chao did was also tested on a second behavioral paradigm. So here what we did is we did a chronic stress. So this is restrained stress where the mouse cannot move, they're immobilized. And then after that, we have fear conditioning where the mouse is listen to auditory cue and then receive a foot shock. And then afterwards, we give them again chemogenetic ligand or psilocybin, and then have an extinction section. And it's known quite a bit by a number of studies that if you stress it ahead of time, you can reduce the amount of fear extinction. So the mouse tend to extinct less, and this is a long-lasting effect due to the stress. So they remain very sensitive to that fear memory. So here we look at what's the effect if we try to silence the PT neurons, and you can see that the mouse learns similarly in both groups. Psilocybin does have an effect, where in the extinction session 1, you can see a notable decrease in the extinction. This has actually been shown by a couple other groups already with other kinds of psychedelics, I think LSD, DOI, that it has this effect on fear extinction. What we tested though is the effect of the silencing the PT neuron, and when you do that, you do see that the extinction curves now really come together. So there is also an effect when you silence the PT neuron, which also seems to really reduce this psilocybin's effect on fear extinction. And we can also look at that on the extinction day 2, which is 1 day later. As well as extinction session 3, which is about 2 weeks later, and you can see similar kinds of effect, although over time it does diminish. So this suggests that whatever we see with silencing the PT neuron, not only is it important for some of the acute stress assay, but even for more chronic stress assay, you can still see the changes. Well, one final behavior that we tested here is head twitch response, which is not so much a stress-related behavior, but more of an acute response to the drug, which I've shown before as well. So here, again, we give the DZZ before the drug and then record head twitch right after that. In this case, we see that in both cases, actually, you can't really change the head twitch. So we can silence the PT or IT cell, and the mouse just continue the head twitch. So some behavior does not seem to be affected when you silence these neurons in the frontal cortex. So what I've shown you so far is that we think that these frontal cortical PT neurons are very important for mediating some of the long-term effects. So this includes some cell type specific structural plasticity. Again, the PT cell seems unique in potentiating the spine size. The PT cell also seems to be essential for the effects on the stress-related behavior. But we were very curious also in terms of what makes the PT cell different in this drug response that lead to its importance for the longer term effect. So one of the things that we wanted to investigate is that you see the spines get bigger, and it's really known that calcium is a very important second messenger for structural plasticity, right? You need calcium influx into the dendritic spine compartment in order for the plasticity process to occur. So we decided that we also want to start looking at that short-term phase to see if we can understand what makes them different that lead to those long-term changes. So this is how we do it. Instead of doing structural imaging now, we express the genetically encoded calcium indicator GCAM6, I think this is 6F, into the pyramidal cell. And then you can see this is just imaging at the apical tuft in layer 1, so you can see the dendrite flashing. In this case, it's actually probably from multiple neurons. And if you zoom in, sometimes you can see more widespread signal, right? These are, as expected, based on different kinds of electrical event possible in a dendrite. Sometimes you see more widespread signal, maybe these are NMDA spikes or calcium spikes. Sometimes you see more localized signal. These are presumably subthreshold activation of the dendritic spine that doesn't spread. And then we can quantify these things. So what Ling Chao did is she imaged, in this case, for 2 sessions before the drug administration. And then we waited for 30 minutes, and then we imaged another 2-field view of the calcium signal. Here we go back to the sparse labeling approach where we're using the retrogradedly transported virus to label the PT and IT cell. And you can see on the right here, the GCAM6F can express very well on the dendritic branch, including visualizing the individual dendritic spines. And what Ling Chao found is that here the PT cell is also uniquely in showing potentiation in the synaptic calcium transient in the dendritic spines. So these are again the spines up at the layer 1. What I'm showing here is you can look at the saline as a pre and post, and then the psilocybin pre and post. So we're tracking the same spines, right, before and after. We can use automated algorithm to extract out the events, right, each of the transients. And then when we count them up and look at the pre-post difference, you can see a clear rightward shift, increase in the number of calcium events in the dendritic spines for the PT cells. Whereas for the IT neuron, we couldn't detect any change in the number of calcium events in the spines. So I don't have it here, but we can do a similar analysis, not on the spines, but actually on the dendritic branch up at the layer 1. There we also see, for PT cell, we actually see a rightward shift, increase of transient in the branch itself, but not for the IT neuron. So it seems like the elevated calcium signaling, at least up at the layer 1, is also quite selective in the PT cells. Okay, so this is where we are now. So I show you that PT cell had long-term effect, and we think that dendritic spines up the calcium signaling is probably quite important. That might then associate with some of the long-term changes. At this point, one thing that we keep asking is, okay, if you get more excitable up there, does the cell body itself also fire more? Because if you remember previously, I showed you in the very beginning that we were guessing going in that the IT cell might be more relevant just based on the receptor abundances. So one might have a couple of scenarios possible, right? It could be that the dendrite is more excitable, leading to also more excitable cell that fire more. The other possibility is maybe there's some distribution of receptor where the dendrite get excitable, but the cell remain about the same, right? Or maybe even suppress. So we want to distinguish those possibilities. And to do that, Pasha Devodian did neuropixels recording. So here, we're not doing imaging anymore, but we're using neuropixel to record in vivo electrophysiology in het-fix mouse. These are awake animals. We go back to the FESF2 and the plexin D1 mice, and then we inject virus to express tranrhodopsin in these neurons. And then we would record for 30 minutes before, infuse a drug. We do it through a catheter so the animal doesn't move too much. And then we record for 90 minutes, and then afterwards we did an opto-tagging experiment where we would shine light, and then if the cell respond, then you know that it's either a PT or IT cell. So how the opto-tagging look like is this. On the left is one example PT neuron that Pasha can confidently tag. You can see that during that time period where the laser is on, you can really see the neuron respond quite vigorously. I mean, the thing to look for is not so much all the spikes, but really the short latency where it can drive the spiking. Actually, as expected, and we see Pasha see this quite often too with the PT cell, they are quite recurrently connected, and you see that you can drive quite a bit of spikes. Whereas for many of the IT cell, it's a little bit more sparse, and here you can see that too. But it's still, you can still quite confidently tag these neurons in the brain. So with this approach, Pasha then asks again, comparing pre and post, which of the cell type might be driving? Are the spiking itself changes? And what he found is that with many of the cells on the left, yellow is the tagged neurons, and then the gray is the untagged ones. On the right, purple is the tagged IT cell, and the gray is the non-identified one. You can see that for the PT cell, most of them are above the diagonal. I think I need to replot this so the yellow dot come out in the gray. We should also change the plot, but trust me that they do on the whole increase the firing rate on the whole. Although you do see some heterogeneity, right? It's not like every single one is driving strongly. But on the whole, they do increase their firing rate. Whereas for the IT cell, there's not a significant difference before and after the drug administration. Okay. So in total, basically what I told you in the second part is that following on some of the early experience, we're very curious about the different cell types. We think the PT neurons are very important for the long-term effects, both the structural and also the behavioral effects. And we think it's associated with also some of the early on selective changes in calcium signaling up at the layer 1. And then we also find associated spiking change in the PT cell in our awake animal. So then that kind of begs the question, right? Why is the prediction different than what we observe? So that's something that we think about now, and hopefully with some ongoing study, we can figure out why that is. There are several possibilities that we entertain and want to really test different alternative hypotheses. They might also work together, they're not mutually exclusive. One idea is that at the end, the psychedelics is not serotonin. So when psychedelic bind to the receptor, they could lead to different ionic current changes, different changes in the memory potential. This could definitely explain why our prediction based on some of the old study, but mostly based on serotonin, might not predict very well what the psychedelics do. But there could also be some local circuit effects. So my favorite theory, but it doesn't mean it's a correct one, we have to test it, is that maybe some of the interneurons are involved. So previously we found that with ketamine, some of the interneurons could be quite important for their effect as well. So it's possible that, for example, if psychedelics act on some of the GABAergic neurons, maybe ones that target dendrites, like the somatostatin expressing neurons, and then if those interneurons also have preference towards PT cells, then that could provide that preference that we observe, right? Even if PT cell is not the one that directly being targeted, through this preference for the interneuron, that could be why we see this bias on their responses. And then finally, a third possibility, and there are more too. I have two more if you want to talk more, but I'll stop at the third one. We have to really test these things out. The third one that I think is also interesting is maybe from long-range inputs. So I think it hasn't been really well mapped out for the frontal cortex as far as I know, but it's really conceivable that the PT and IT cell can receive different types of long-range inputs. And then depending on how the drug acts on these long-range inputs, there could be activity-dependent plasticity and also activity-dependent responses that might be even independent from what's happening in the local circuit, and in conjunction is what select for these cell type specific responses. Okay, so to conclude, I show you some evidence that psychedelics can induce plasticity in the dendrite structurally, and then also in terms of gene expression and brain-wide. And then on the second part, I told you about some new data doing some circuit tracing that really highlight how these cells can have really cell type specific effect, particularly pinpointing the PT parameter cell being quite essential for some of the behavioral effects of the drug. And then I do want to go back to the initial question where the lab, we're very interested, like once we identify a circuit, we really want to go back and ask how may we eventually use some of these information and target these circuits to engineer better treatment for depression. So that's the question I'll leave you with. Thank you.